All right, we have CRISPR. So we're starting off with looking at our absolutely amazing molecular scissors. So these are very precise scissors. This is an enzyme that we can use to actually generate amazing cuts in the DNA exactly where we want it to be. But the thing is, it existed as something from bacterial cells originally. So CRISPR-Cas9, an absolutely amazing enzyme that will cut DNA exactly where we want it to cut. Our study dot point that we need to look at is the function of CRISPR-Cas9 in bacteria and the application of this function in editing an organism's genome. So we need to look at Cas9 and how it works in bacteria. And then we need to know how we have modified its process to work in our cells and to be able to cut DNA in our cells. Because remember, DNA is universal. So whatever happens to DNA in one cell can happen to the DNA in another cell, which means if we have a mechanism that can cut DNA in one cell, we can transfer that and use it in another cell because the DNA is exactly the same in every single cell. So CRISPR actually stands for clustered, regularly interspaced, short palindromic repeats. We've already mentioned palindromic repeats when we looked at our endonucleases, right, our restriction enzymes. So we kind of looked already at palindromic repeats. They're those ones that repeat both ways, so the word that can go forwards or backwards. The rest of it we'll start to look at as we go through. So clustered, regularly interspaced, short palindromic repeats. You'll understand what this kind of means as we work through this. So you have the bacteriophage that we talked about the other day. This is our typical bacteriophage, which is the enemy of any bacterial cell. This is the virus that will infect them. So these are their, sworn enemy that we don't want to have to deal with. Our CRISPR is actually our bacterial cell's immune system. It's our bacterial cell's way of providing a defense against the bacteriophage. And we did talk about this briefly the other day. So I've already said that, phage for short, if you want to shorten that. They attack, infect, and kill bacteria by injecting their DNA into the cell, hijacking the system, and then they break apart the bacteria once they've been built. So they escape by just breaking the bacteria. So of course that lysis of the bacteria means that the bacteria is dead and cannot continue to function. So they do just kill the bacteria basically. Yes?
So CRISPR just happens naturally?
Yes, not in the body.
Naturally in cells?
Bacterial cells. Yep, doesn't happen in our body. We don't have CRISPR in our body because we don't need to defend against a bacteriophage. Now, the other interesting thing that I read a few years back is bacteriophages being used as a particular target to kill some of these bacteria that are infecting us now, like golden staph. We can start to use a bacteriophage to target specific bacteria and kill them in the body without harming us because a bacteriophage doesn't harm us. So, interesting. It can be used both ways. All right, this is the infection of a bacteriophage into a bacterial cell. So it comes along, it will attach to the bacterial cell. So it will use its little legs, and it's kind of like a lunar landing module. It comes along and it lands on the bacteria. Its legs attach and then it injects. So it basically is like an injection that it sends the DNA into the cell. Of course, remember our bacteria does not have a nuclear-bound membrane. So if it injects that DNA into the cell, what's going to happen? No?
It's going to make the proteins kind of...
Yeah, what's going to happen to our piece of DNA?
To accept it into the genome.
Nope.
It's going to be made.
Translation and transcription is going to occur. So because we've got no nuclear membrane, we can just inject that DNA into the bacterial cell and off we go. Our DNA polymerase will latch on and away it goes. Transcribes the messenger RNA. Then transcription's occurred. Hang on, did I get that around the wrong way? Translation, then transcription. Translate first. Copy the recipe. Translate it, then hang on.
Transcribe.
Oh crap.
Transcribe. You can't copy a recipe that you don't.
You're right. Transcribing is copying. Translating. I hate days like this. Yes, I get the two confused, and I know what I'm talking about.
It's some Eastern European lady's recipe.
Yes.
For something like
yes.
or something, and you can't figure out what it says
until you turn it into English.
And
There you go.
Translate it into English, and then to preserve the document, you need to transcribe it into something else.
But is it the transcription?
Transcription comes
Transcription,
first.
whatever. Okay, it's not my problem. You get the
Love
order
it.
wrong.
So, what comes first?
Transcription, because it C's before L, right?
It's transcription first.
Yes,
Transcription
C is before L. Yes, that's the other way. That's the douchey way to remember it.
So you
There's
preserve,
if you've watched-
preserve the document and then you translate it.
Yep.
There's new technologies that can help, that are helping people make custom bacteriophages.
Yes,
Planning on it.
yeah, it's,
I love it.
there's lots of things that we are so that's why biology is such a big field, because you can narrow it down to any particular area and start to really research into any particular area, and it's just amazing. All right, so our DNA comes in. It's transcribed, then translated into a protein. All right, that protein happens to be the bacteriophage. So it creates the capsid, it creates the little tube, it creates the injection, it creates the legs. So it's creating all of that inside the bacterial cell. And you can see, look at the bacterial cell down the bottom there. It is full of phages. Once it gets too full of the phage, it then lyses, and those phages escape and continue to attack new bacteria. So it's exactly the same process as a virus would infect us. Same sort of idea. So this is the bacterial cell's mechanism for fighting against these bacteriophages. If our bacteria survives the infection, they defend themselves from future infections by chopping out a bit of that invading DNA. So they save a bit of the DNA, and they save it in their DNA strand. So they can then identify the foreign DNA next time. So it's like they've kept a teeny tiny little bit of a copy of that DNA in their genome. And so next time it comes along, they can recognise that little tiny piece of DNA when it injects, and they go, oh, don't want you, and they destroy it before it starts to be replicated. So this is the amazing part. This is where they add that piece of phage DNA into their genome. So it'll take that little piece and insert it into its own DNA genome. This is how it does it. It grabs the phage DNA that it's injected in there, and this is second time round, of course. All right, so this is second infection of the phage. It's already got that piece of DNA. Well, this is where they have survived the attack, and they've grabbed that piece of DNA and they've added it into their genome. Can you see their genome has 4 different colours? They're previous phage attacks. So each time it gets and survives a phage attack, it inserts that DNA into its genome. Of course, when it replicates through binary fission, what does it also replicate? The DNA and the phage DNA. So it's keeping that copy every time it replicates. It doesn't have to find them all new. It's keeping that copy every time it replicates. Question over?
I have 2 questions. Firstly, if it's copying the phage DNA, what protein will that actually code?
It doesn't code for protein.
Okay,
We'll
so
see what,
it doesn't really.
It's just copying that piece of DNA. That's not a coding section of, and we'll get to what that section actually is. We'll look at exactly what this is. So this is where the word CRISPR comes in, because you can see there's repeats here of stuff. So this is where we start to narrow down what CRISPR is. So good question. That leads us into what we're looking at now. Yep. We'll get to it. Hang on. We've skipped ahead.
My other question is the way that DNA kind of like keeps it, not DNA, the way that bacteria kind of keeps it up is because they divide.
Mm-hmm. And they replicate their entire genome, don't they, when they divide?
Yeah, they do. So it just keeps it. So
yes. So it stores it indefinitely in its memory of DNA.
So all the bacteria will have it.
So each time, yes, that whole colony of bacteria will have those particular phage DNAs stored already in its memory. So it means that it doesn't have to find that DNA every time. It doesn't have to get a new infection of phage DNA, because, of course, if you did that, could you imagine you would likely die? If you had to protect yourself against every phage every time, the likelihood is that at some point in time you would die. So it's able to transmit that information on to the next generation by binary fission, because, of course, it's replicating the DNA as it replicates for binary fission. All right, our CRISPR locus. So this area that you can see on the screen there, that phage DNA, is known as our CRISPR locus. So this is the physical location where it stores that phage DNA. And this is where it starts to get into our CRISPR process. It has a very characteristic structure. The phage DNA is separated by sections of repeated DNA sequences. These are referred to as our spacers. So you have a piece of phage DNA, then you have a spacer. And that's a repeated sequence of DNA. And then you have another phage, and then you have another repeated sequence of DNA. So you can see that we've got phage, repeat, phage, repeat, phage, repeat. So we can see these CRISPR locuses have constant repeating through. So this is where we come into the CRISPR meaning. The origin of the name of CRISPR comes from that locus. The DNA is incorporated between clustered, so they're all clustered together, regularly interspaced, short palindromic repeats. All right, so clustered, regularly interspaced, short palindromic repeats. That's this.
It's going to be the new Mr. SML.
So we've got clustered, regularly interspaced. So have a look. The spaces between them are regular. Short, they're very short palindromic repeats. Those spaces are very short palindromic repeats. Does that make sense? Yep. Yeah, it's pretty cool, isn't it? Pretty cool. So each of these repeated DNA sequences spaces out. So these are basically your dividers of your binders, dividing up each of those phage DNA sections. And they're found at regular intervals. So we know that we keep a particular section of the phage DNA, and it should only be X number of base pairs long. And then we have a little repeated section, and that repeated section is the same, and it's palindromic. And so the cell knows that repeated section, and we can get to how it manipulates that. So the letters of the repeated sections, you don't need to know this. G, 4 T's, 4 A's, and a C. Guess what? Run that backwards. What do you get?
Good talk.
Good and talk. Exactly the same. C goes with G. Yeah, good and talk. C goes with G. It is GTAC. Why do you think GTAC has the initials GTAC? Mind blown.
That's such a biology.
It is a very biology nerdy thing to do. It's like have you seen that car that looks like a square box? There's a car that looks like a square box.
Yeah, the cube.
Yeah, the little cube, right? Yeah, it is called a cube.
Yeah.
I found someone with the number plate C6H1206 on their white cube.
Oh.
It's a sugar cube. And I went, oh my God, I love it. C6H1206. Glucose.
I have an idea yesterday, and I think I should do this when I got today. I was thinking, well, to make everyone happy, or more to myself, every biology lesson I can use a biology related quiz.
Very fun.
What kind?
Yeah, go for it.
Who will I pick up? Some plants?
Who knows? Yeah, there's plenty of them. All right, so can you see here? This is our little memory. This is our memory function for our CRISPR. So CRISPR has already got the DNA from the yellow one, the DNA from the green one, and the DNA from the blue one. So if any of those phages inject their DNA in there, we can recognize it and we delete it basically.
Ready for you.
Oh yeah, it's so cool. So you can see in between the phage DNA, we've got those repeated sequence, the GTAC. All right, so GTAC is repeated in between each one of those sequences. We then use that to create our CRISPR RNA. So this is where we start to get into our nitty-gritty details of the fact that that CRISPR locus, so that piece of DNA that you see, is constantly being expressed in the cell. So it's constantly being made. It's turned on all the time. Think of like a constituent gene. It's always on, all right? Always being produced. So transcription occurs to produce the CRISPR RNA molecule containing the identification of those phages. So it's got that phage DNA in it. This is what we call our CRNA, so our CRISPR RNA. We
Sorry,
also
I spent way too long trying to draw the 2 slides ago.
That's all right. You can always listen to the recording back later. So our CRISPR RNA is pretty much our copy of, okay, if this gets in, if this gets in, if this gets in, we can find it. So you can see here in the little picture over the side, we've got our CRISPR RNA, but it can't work by itself. It needs to have something with it to help it out. Because just that piece of RNA by itself, what's it going to do? It can't do anything. It will create a protein if you could try, but you don't want to create a protein out of it. You want it to defend the cell. So it has to work with another complex. And this is called the trans-activating CRISPR RNA, or the tracer RNA. And this attaches and binds to the sections of the repeated bits of the CRISPR RNA molecule. Now, if you have a look at my very tiny picture over here, you can see that we have our CRISPR Locus DNA, where we've saved the phage DNA into our CRISPR Locus. We then transcribe that into our CRISPR RNA, so our CRRNA or cRNA. We can then put that together with the tracer. So the tracer is this section inside, the sort of the orange section there inside. That's our tracer RNA. We combine that with our CRISPR RNA and our Cas9 molecule. So the Cas9 molecule is an enzyme, and so we'll get to that in a second.
Moving quite fast.
Okay. Is that because you spent too long on the slide though?
No, but like in general.
Okay.
No fear of saying it.
No, but I'd prefer she says it, so I know to slow down. If
It's
people,
my learning.
yeah, if people,
I think it's a good thing.
Yeah.
Never.
but my frustration is that people often don't speak up. They just sit there and assume and just, you know, whinge later. I'd rather you whinge now so I can do something about it and fix it, rather than whinge later and, you know, whatever. Never hear about it. Never know.
So the tracer activates CRISPR.
So back here, we've got our CRISPR RNA. That's the RNA that's copied off our CRISPR locus. So that's that little strand where we transcribe it. We create an RNA molecule that has the sections of the phage in it. So we've got the CRISPR locus where we've copied the whole thing, and we've got phage 1, phage 2, phage 3, and phage 4 spaced with our GTACs. So that's our CRISPR RNA. And we call that cRNA or CRRNA, either way. You'll find it different in different books. Then we have to combine that with a tracer, because the CRISPR RNA by itself can't do anything. And you'll see why in a minute. So we're just introducing the different terms at the moment, but it doesn't make sense because we haven't seen how CRISPR actually uses this. So what happens is we combine the CRISPR RNA with our tracer, and that combines into our CRISPR molecule. So that's an enzyme. That big fat jelly bean looking thing is actually an enzyme. So that's a protein that the cell, the bacterial cell makes, and then it produces the CRISPR locus, produces that.
Locus is just location.
Pardon?
Locus is just like location.
Yeah. Yep. So that's where all the phage DNA gets stored. So that location where all the phage DNA gets stored, it transcribes that into RNA. That RNA is what, instead of calling it messenger RNA, because it's not a messenger to go and be produced by a protein, it's a CRISPR RNA. So it's still RNA, still kind of does the same thing, but this time, instead of going and producing a protein, it's going and locking into the CRISPR molecule. So it has to lock into that tracer. The tracer fits into the CRISPR molecule in a particular pattern. So see how it's got lots of little loops on the tracer? Yeah. Those loops fit into the protein molecule. So they bond with, let's say, hydrogen bonding, or it could even be, you know, disulfide bonds or ionic bonds, whatever it happens to be. They fit into the CRISPR molecule. So remember, the CRISPR molecule is just a protein. You've got primary, secondary, tertiary, that sort of thing. It's just a protein. And we've got our tracer that fits in perfectly. So our CRISPR RNA connects to the tracer, and the tracer fits in place into CRISPR. So this is what forms CRISPR-Cas9. So Cas9 is the term for the enzyme. Now we have different CRISPRs that we're finding at the moment. This is a very new field and there's a lot of research going on at this time. Actually last year, because this was the first time that I had come across CRISPR as part of the course, it hadn't been something that, other than seeing it in the news and reading articles on it, I hadn't really done much about it. So there was a course over in Europe being offered online at 2 a.m. in the morning for me, which I attended for 4 weeks. And so every week there was a class on where we got to meet some of the scientists who were leading the field in this area. And we also did some homework tasks and a few other bits and pieces to actually go through the course. And I did that course, and by the end of that course, holy moly, the amount of work that they have done with CRISPR and what they can do is just incredible. So if you do get around to watching the Natural Selection on Netflix, you'll understand a little bit more about what's going on with this field. It is a very amazing field, and if you ever want to get into this sort of area of research, it is just mind-blowing what we might be able to do. All right, so we've got our CRISPR-Cas9 system. So remember, Cas9 is just talking about the enzyme molecule that we're binding to. Our guide RNA, this is the bit that we've produced, our CRISPR RNA. This makes a guide. So we use that CRISPR RNA to give us like a target to connect on to our invading phage. Now we'll explain how this works in a second, but just understand that we've got our CRISPR Locus, we've transcribed it into our CRISPR RNA, we've inserted the CRISPR RNA into the tracer, and that has then locked in place into our enzyme. I'll pause there.
I think the textbook calls it like sgRNA. So in my notes, I just got sgRNA and then CRNA.
Yeah, that's single guide RNA, I think. There's lots of different names for this stuff, but yeah, you're basically looking at, you've got your guide RNA or your, you've got your tracer, you've got your CRISPR RNA, which when linked together will be called the guide RNA. So when you link that with the tracer and the CRISPR RNA, you get the guide. But before we get the guide, we have to chop off the other bits that aren't needed, because of course we've got 4 different phages connected to this. So there's another we'll get to that.
But it's a CRISPR RNA plus tracer RNA.
Is your guide RNA. So once you've got CRISPR plus tracer, that's your guide RNA. So we will work on this. Remember with the operon how we just threw all this stuff at you and then eventually it worked in place? The same sort of thing is happening here. We're throwing all this stuff at you, then we'll get the models out and we'll start to work on how this actually works, right? Because we've got the lovely little models for this.
What does the tracer RNA actually do? Like it binds to the...
It binds the phage DNA that's invading.
Oh, yeah.
So it binds to the phage DNA and cuts it.
And cuts it and puts it in there repeatedly.
Yep. Well, it doesn't need to because it's already found it. If it's in the guide RNA, it doesn't need to insert that piece again.
Oh, okay.
It only needs to insert that piece when it's first infected with a different phage, one that it hasn't seen before.
So, phage comes in, tracer RNA is like, dude, what the
I
hell?
recognise you.
I see you, you're not meant to be here. Grabs him, cuts him.
Yep.
Wait, tracer or guide?
Tracer.
The guide RNA is just to spot the.
And then what happens? How does it get into that little CRISPR?
That's what we're looking at.
So that's what the CRISPR...
That's what we're about to look at.
Ah.
How the enzyme works to cut it.
I'm
and
great
then
at leading into your next
you
slide,
are.
aren't
You
I?
all are actually.
Wait,
You
so-
all do this. It's like, but hang on, what about this? It's like, well, we're just about to cover that.
Wait, so the tracer RNA actually just
No. We get to the cutting. The enzyme does the cutting.
Yeah,
All right, the enzyme does the cutting. So Cas9, this is where we look at Cas9. So this is where we look at our nuclease. And a nuclease is an enzyme which can cut DNA. So it's very, very precise the way it cuts DNA. It's nice work. It's guided by the information encoded by the CRISPR RNA sequence. So remember how our CRISPR RNA sequence had different colours in it from different phages. It's guided by that. And it's looking at the particular section. Now note that really long sequence of CRISPR RNA is actually cut into little sections where it needs that phage or that phage or that phage. And it cuts it based on those palindromic repeats. So it knows if the palindromic repeats there, it chops at the palindromic repeats and it's got the phage DNA in there. All right, so that's why it intersperses with those palindromic repeats, because we looked at endonucleases before and we looked at the fact that they cut with the palindromic repeats. Okay, they can either cut blunt or sticky. Now you don't need to know how these are cut. What you do need to know is that each one of the repeated sequence will be cut, so you then grab that phage DNA. That phage DNA is then your guide. So that phage DNA is the bit that it's looking for. And so we'll get to this in a minute. So we're jumping ahead a little bit.
Tracer goes,
Yes,
and
it recognises.
It goes CRISPR CRNA plus tracer RNA makes guide RNA.
Yep.
And then that guide goes, hey Cas9, goes to Cas9, Cas9 goes, wah.
If it finds the phage DNA. Yes. So the guide RNA gets that structural support from the tracer and it's able to lock into place in the Cas9 complex. So it binds to the complex and directs or guides Cas9 to find the enlarging phage DNA that matches that sequence. So it's already got a teeny bit of that sequence in it and it just goes along and it looks for that sequence. Now there are secrets to how it looks for that sequence because it doesn't just scan an entire long section of DNA. Why are you looking at me strangely?
My brain just hurt.
Okay, it's in learning mode, that's why. It does hurt in learning mode.
I think it's uncomfortable.
I know.
It actually genuinely hurts.
Yes, it does. But the other factor of it is that your brain, and this is what we learned in the staff days. No, in the staff days, we learned, a guy had come in to talk to us about how the brain actually functions in terms of neuro-linguistic learning. And he was talking to us about he calls it top-down and bottom-up mode. The top-down mode is where you're learning new stuff. So you have to really concentrate. Your brain is really working hard and you've got about 3 hours worth of energy in that mode. That's it for the day. This is why we have to go and rest. This is why we have to go and sleep. The bottom-up mode is just building on what we've already got. So we're building on information we've already got. So if you're learning something and you're going out and you're driving a car and it's a repeated process and you're learning a little bit more and you're learning a little bit more, you're not in that top-down mode all the time. You're only in the bottom-up mode and you're just tacking a little bit on the top every time. All right, so there's different ways that we learn, but right now this is all new information, so you have to be in top-down mode because you've not seen this before. So that's why your brain hurts, is because you are actually, using it a lot, and you'll feel tired afterwards because you've used a lot of energy. You've got 3 hours worth of that energy to use. So this is why you have recess and lunch, because you have those breaks in between. You can recharge, you get food, you can rest. Your brain just isn't.
But if we only get 3 hours for the whole day, why am I expected to do 6 periods of class?
Yeah, that's our problem. And then you look at those kids that he was talking about in the assembly the other day, where they're learning from, 5 in the morning to 10 p.m. or 1 a.m. in the night. I'm like, whoa, that's not feasible for a human brain to do. Not feasible. It's just like, whoa. Okay, back to this. We've got our search and destroy function. Yeah, I know. That's terrible, isn't it? Search and destroy function.
That's actually interesting.
It is really cool. Cas9 is just...
Does Douchey have a video?
Wow. Yes, he does. Yeah. Yeah. So Douchey has a video, and there's also on Infintina, I've got Douchey's podcast as well. So they're like an hour long, some of those podcasts. But if you're traveling on a train or you're just in the car and you go, Mum, sorry, I'm listening to biology stuff, can't talk now. You can listen to those. And I don't care. If your mum wants to listen to it, great. I don't mind. But they're really good to listen to as you're traveling somewhere. Oh, God. Okay. Back to this. Cas9 complex is now ready to find an invading phage that matches the DNA sequence in the guide RNA. Notice the picture has a smaller section here. So have a look at this. This little part here, let me get my little laser pointer up for you. Why is my mouse being a pain? You've only just realised the year 11s aren't here.
No. I said they're not going to be able to see the laser pointer.
I was getting the laser pointer up on my screen, but when I do that, it goes back to minimised mode. Like this. So see this bit here? See that bit there? That's the piece of phage DNA that it's looking for, all right? So see how it's shorter than it was before? Remember that piece was really long with different colours in it?
Yeah.
It's cut it off. It's now, pardon?
Never mind.
It's produced a CRISPR that is specific to that phage. And of course, all those other little palindromic repeats, it'll cut them off and add them to a tracer as well to produce exactly the same thing. And remember, this is happening constantly in the cell. It's not an on or off mechanism, it's always on. So it's always producing CRISPR molecules with those sections of guide RNA. So anytime that phage DNA comes in, the CRISPR molecule unwinds the phage DNA, checks whether or not the guide matches it, because remember, that's RNA, checks whether the guide matches it. If the guide matches, if the hydrogen bonds link up, chop. Chops the DNA.
It's cutting off the...
It's cutting the
dark-
phage
wait,
DNA.
It's cutting off the dark purple in that little separated bit.
Both of them.
It's adding on the light purple, the light pink.
No, the light pink is just a tracer. To trace, that's correct.
So this part here is the guide that locks on. Here is the phage DNA.
And that's the tracer plus the CRISPR RNA.
This is the CRISPR part. That's the CRISPR part with the tracer attached to it.
Wait, so orange is tracer.
Yep, orange is tracer,
And pink?
and the pink is the guide RNA. Okay, that's your CRISPR RNA. That's your guide RNA there.
Yeah.
This is the phage DNA here, right? So you can see the little enzyme around here. See how that unwinds the DNA?
Yeah.
So it unwinds the phage DNA.
Yeah.
It links up this piece to the phage DNA.
Yeah.
If the hydrogen bonding matches,
So like if it's got the complementary
yep,
vector,
it cuts.
and it cuts,
There and there, both strands.
so that piece of guide attaches onto the phage.
And it cuts both strands. The guide detaches
Yeah.
and the strands are now cut in half.
And then
They can't join back together because DNA that's cut in any cell is dangerous for the cell, poisonous for the cell. So the cell actually destroys DNA that doesn't have any protective ends on it, destroys it.
So this is how it like...
Yep, this
So this is
destroying the phage DNA that's got
this
into
is
the cell.
After it's got the memory.
Into the, yes, after it's got the memory.
Putting it into the memory.
No.
we already did that.
No, that's the first few slides.
Yeah, no. Okay.
Yeah. So this is already in the memory because, of course, remember the CRISPR Locus already has that all in there. You've got the 4 different colours in the CRISPR Locus. So we just copy that and then we grab all 4 colours, we cut them up and we attach those to different CRISPRs. So we've got 4 different CRISPRs, one that will go orange, one that's green, one that's blue, one that's purple.
So let me talk you through it to make sure I've got it right.
Yep.
We see a piece of phage in our DNA, in the bacteria.
Yep.
And it's like, dude, no, you're not meant to be here.
Yep.
So it takes a little section of it.
Yep.
And it puts it in its own DNA.
Yep,
in
in the
little section,
locus. Yep.
In the
CRISPR.
clustered, regularly interspaced,
Yep.
short palindromic repeats.
Yeah.
Excellent. Nice work.
So it's put it in there. And then it's like, and then, I don't know, some time passes, and it sees that piece, that phage is like twin brother or something. And it's like, I actually just kicked you out. What the hell are you doing back here?
Yep.
So it takes it, there's an enzyme that unzips part of that phage's DNA, and then the guide RNA, which is, it's got the tracer and it's got the CRISPR RNA, so CRISPR plus tracer is equals guide. So the guide RNA attaches on, and if it's got them complementary base pairs, which it knows because it has that memory in the locus, then it's like, oh, yeah, no, I did. So you are cut off. That's pitch perfect reference.
Yes!
So it cuts it, but then because it's cut, the bacteria cell is like, whoa, we do not appreciate that toxic negativity in here.
Exactly.
You are cut off, out you go, and
Yep.
it destroys it.
Yep.
And then it's protected against the phage.
Can we do that more often?
Well done.
Yes,
That is exactly right.
I
Perfect.
understand things.
Perfect. Very nice. That's actually really good. Really good. Well
That
done.
helped me.
So our,
I get it.
as we've just discussed, our Cas9 complex comes along and cuts that invading phage DNA. It inactivates it so that phage DNA can't now hijack the cell. Because if the DNA is cut, It can't be transcribed and translated, all right? If the DNA is cut, as we discussed, it's extremely toxic for the cell, and the cell will actually start to destroy that piece of DNA. So it'll just break apart the nucleotides and reuse those later on, all right? So it'll actually borrow those nucleotides to use for its own purposes.
Okay.
So this is its way of protecting itself. Now, because of the fact that it has DNA as well, how do we stop the Cas9 complex from accidentally cutting itself? Because what happens if that particular site matches something on its own DNA? The CRISPR-Cas9 molecule will come along and it will cut it.
Metal groups.
Yes.
Oh, like it has to stick out, so
It's got lollipops.
All right, so
yeah.
the other factor of it is there's a particular site. This particular site is called the PAM, the protospacer adjacent motif.
Pam holds the lollipops.
No, PAM doesn't hold the lollipops. PAM is a recognition site. So that protospacer adjacent motif site is the part that it's looking for. When the bacteria obtains the section of DNA from the phage, it doesn't put the PAM site into the code. Now this becomes important. Now we'll get back to this in a little bit. So that PAM site is a very, very particular site, and our Cas9 complex looks for that PAM site, but it doesn't actually insert the PAM site into the guide. Right? So the PAM site is left out of the guide. So when it cuts the guide, it's not putting the PAM site into the guide. Right? It leaves it completely out. Now we come back to the PAM site, so just hold your thoughts on that one. All right, these are the two ladies that have done an amazing amount of work. Yep, these are the two. So Jennifer Doudna and Emmanuel Charpentier worked out a way to modify the bacterial guide RNA into one piece of RNA that could be synthesized in the lab. So they didn't discover it for starters, right? They worked on some work that another person had worked on. So we all think that they discovered CRISPR. They didn't discover CRISPR. CRISPR had already been discovered, but what they did was work out a way to modify that guide RNA. Now that's important, because if we can modify the guide RNA and we can tell it what we want as the guide RNA, wow, we can now program it to cut DNA exactly where we want it to. Yes, massive bioethical issue. Hello. Why?
Like natural.
We're altering someone's DNA.
What if someone and then they would then use this to add a bit of heat in your body so you make them really smart? It's unfair for
We get to this. We've got a slide on this. So we get to this. So by combining the CRISPR RNA and the tracer RNA together, they formed what we call the sgRNA, so single guide RNA that can combine with our Cas9 molecule.
This is why we can't have nice things.
I know, this is why we can't have nice things. We can now program Cas9 to cut any sequence we want. Scary. So we're going to run out of time, unfortunately, but I'm going to introduce the funky B gene.
The funky bee.
The funky B gene.
You know what I think I need? I think I need CRISPR to insert whatever like soul's brain to like come in here. So
The funky B gene, when it's mutated, there is a loss of funk.
No!
Oh no.
Never die.
I know. The funky B gene in this image has a mutation. That purple bit in the image is a mutation. Okay, so you might want to highlight that little bit. Just highlight any of them. Doesn't really matter. But something in the middle. Highlight one of those. So our funky B gene has a mutation. We form our Cas9 complex with our fantastic
funky bee.
new guide RNA that we have produced in the lab. Hello, hello. First off, we have to identify the mutation. So we need to know where that mutation is. And often it is one base pair that has mutated. So we need to run that person's genome, find out where that issue is, and identify it. We then need to produce a guide RNA that has the same sequence as a funky bee gene mutation site. Cas9 comes along, matches it up, and cuts the DNA of both strands of the DNA at that site. This occurs, and this becomes important later, 3 nucleotides downstream from the PAM site. Remember we mentioned the PAM site just before? That PAM site is not included in the Cas9 locus. 3 nucleotides downstream from that is where your CRISPR-Cas9 cuts the DNA. We'll come back to this. The DNA, as I said, is toxic for cells, so they try to repair the break. Now, they come around and there's 2 ways to repair it. One is very error prone. They just come along and try and reseal it by adding more nucleotides. So, this is very error prone. The gene probably won't likely be restored properly. So if we want to make sure that gene is properly fixed, we add some donor DNA in there. So we put in the Cas9 complex with our produced guide RNA, and we also put in a piece of donor DNA that matches the sides of the break, but also has the right nucleotide in it to replace it. And so what happens is that piece of donor DNA gets inserted basically into the cut site, and it replaces what was already there. And so what we can do is we can replace the faulty gene with the correct gene, and we can repair that function. So this is where we get to the little boy on Netflix who has the degenerative eye disease where he doesn't make that protein. It's 1 base pair that is incorrect, and all he needs is to replace and fix that piece of DNA within his eye, and he is restored.
Can they? Will they?
Well, watch the series. Like, it's amazing. The series is actually amazing. But those poor parents living in the US, can you imagine?
The ethical.
The cost. Massive cost. No. So they had to, it's sad. It's really sad. But they did everything they could to fix their son's sight because they knew that he was going to go blind if he continued to grow without this treatment. So yeah, it's like that whole series is amazing.
Did you put that for your daughter?
Pardon?
Did you put that for your daughter?
I don't know if I could afford it, but I couldn't afford that. I don't know how they did. No, I don't know how they did. All right, ladies, we will come back to this next session, but it is just amazing. CRISPR is one of those things that mind-blowing.